Hi, everybody. Welcome to the Thematic Session 2 with the topic Synthetic Biology and Bioeconomy. My name is Clementine, and I am the MC for this event. So, without further ado, may we welcome and invite Professor King Cheuh, professor at the Division of Life Science, to be the chair and moderator of today's session, as well as giving us an introduction speech. Professor Cheuh, please.
Thanks, Clementine. So today's section actually is something not necessarily new to UST colleagues and friends. We've been doing synthetic biology for quite some time, but then I think that today actually is an interesting section because actually we want to learn more about how this development of synthetic biology in different regions have been taking place. And so today, actually, I'm very fortunate to have a number of colleagues from US, from UK, from Singapore, from mainland China, as well as in Hong Kong. They're going to talk about the development of synthetic biology, or sometimes actually maybe it crossover with biotechnology, as well as how it's really affecting the future development of bioeconomy. I think that with that as a backdrop, I hope that actually we will learn a little bit about different regions, how they are planning with all this development, with the collaboration from the industry, from the university, from the business sector, as well as the government, how they form a good ecosystem that make this particular field bloom. Okay, so with that, I would like to pass on to our MC so that we can introduce the first speaker from UK.
Thank you, Professor Cheuh. Without further ado, let us welcome Professor François Benex, Vice Provost for Innovation and Director at CoMotion, Charles W.H. Matthew Professor of Chemical Engineering and Director at the Center for the Science of Synthesis Across Scales at the University of Washington. His keynote speech will be about the CoMotion Innovation Hub, igniting entrepreneurship at the University of Washington. Professor Benex, please.
Thank you, Clementine, and thank you to the organizers for inviting me. It's a pleasure to be here. Let me go ahead and share my screen, and I will go into presentation mode right now. So let me tell you a little bit about CoMotion at the University of Washington. CoMotion is essentially much more than a tech transfer office. This is an office that is dedicated to really drive impact from researchers from the UW community. And what we really want to do is to nurture and empower innovators that eventually will change the world. I will tell you a little bit about how we do that and what is our approach to do so. I'm a researcher myself, but I will be talking mostly about the envelope that we put around these folks to make them successful and to allow them to create new startups that will make a difference. So CoMotion offers a pipeline of interconnected services, and I like to define it as a one-stop shop for innovation. What we do is innovation training. We feel that this is very important. We want to introduce young folks, in particular graduate students and postdocs, to the concept of entrepreneurship, and we spend some time actually lighting up this spark through a number of different offerings. When we have a record of innovation that comes to the office from researchers, we spend some time talking to them about how these inventions might be protected and to develop a proper strategic approach to intellectual property. We provide funding pre-company to help some of these pre-startup develop their innovation and prototype, and we provide mentoring and networking services, eventually licensing the invention into startups that we ourselves incubate. So these services are actually yielding a lot of fruits. You can see some of the numbers over the past 5 years. We have conducted well over 2,000 innovation trainings. We typically file about 711 patents or so over 5 years, getting about 100 of them issued, US patents only, and we distribute about 1 million of funds per year into our startup, and that yields about roughly 400 licenses or so per year. So I wanted to point out the acceleration that we have seen in the Puget Sound ecosystem, which is in the state of Washington and around Seattle and the University of Washington. Over the years, the University of Washington has started roughly 250 companies. And these companies have went on to gather about $8 billion of economic activity. And you can see from this graph on the left-hand side that about more than half of that, in fact, $4.7 billion, have been gathered over the past 5 years. So it's been a tremendous acceleration. We still have about 100 or so spin-offs that are still active. They employ over 1,000 people in the state of Washington and probably about 1,800 throughout. Most of these innovations come in the therapeutics and device area, including medtech, but also in software, most of it related to health, but some not related to health, and in diagnostics. So this is typically the case for a strong medical school, and we see these numbers in other places as well. I like to use some analogies from the work that I'm doing, which is really at the boundary of synthetic biology, molecular biology, material science and engineering, and nanotechnology. And one of the things that you notice is that growth always happens at the boundary, as in this example where you see, in fact, a scanning electron micrograph of a growing titanium that is spurred by small peptides. And what it takes to really be successful and to have a vibrant ecosystem is world-class researchers, and every single university have those. These folks are creative, they work collaboratively at the confluence of fields, and really they drive innovation. A few of them are highlighted below. We certainly have top quality talent in protein design, computational protein design, with the Institute for Protein Design that is led by David Baker. Jay Shendere has made tremendous contributions to the field of computational genomics, and a lot of these other folks that I showed here, and this is certainly not an exhaustive list, have been contributing quite a bit to our entrepreneurial ecosystem coming out of the University of Washington. If you think a little bit about how a material grows, this is a bit of nerding out right here, but you can look at how the free energy varies when different molecules or different atoms come together. And if you put things together, they will actually increase in interfacial energy, much like you can see right here indicated by the blue curve. And what happened though is that if something grows sufficiently large, exceeding what is known as a critical radius, then the volume free energy will dominate. And what will happen is that the energy will go down, and eventually the sum of these two will also go down, which means that the material will be stable. So I like to compare these two kind of opposite effects to what we have in early research, the innovation development process, and really the market pool, which is what is going to drive this innovation forward. So critically, we get a lot of early stage innovation from our researchers, from our trainees, from our innovators, and these are fragile, and they can dissolve back into thin air, as you will see here in the case of the blue curve. So really they need to be nurtured before they can start growing on their own and actually becoming full-fledged startups. Oops. Another analogy that I will be using is this idea of the protein folding funnel. Some of you in the synthetic biology field might remember this funnel, which was proposed originally by Alan First. And the basic idea is that an unfolded protein, such as this one, can sample a different of conformation. And these conformations are associated with a number of kinetic traps and eventually some folded state. And some of them are going into deeper or shallower type of energy minimum. For example, these amyloid fibrils right here would be at a very low energy minimum. But in the case of entrepreneurship, and when a startup navigate, for example, different product, these wells are much shallower. And what you have to think is how a particular technology needs to evolve and navigate this landscape to find actually the right product that is going to have traction with the marketplace. So really what we want to do is to help our startup create a product that customers eventually want and will buy. So how do we reach the critical radius? I mentioned the importance of developing the innovation mindset. We are certainly keen on doing that. We want to focus on applied or use-based rather than theoretical. It does not mean that you shouldn't be doing basic science, but the basic science can be viewed within the context of a particular practical problem. We like to favor multidisciplinary team because we feel that they have more chances to tackle a particular problem in a more innovative fashion. And we like team-based and experiential type of project like many other places do. So to do so, CoMotion deploys a number of mechanisms. We have a number of just-in-time workshops and training that include tech commercialization, fundamental for startup, idea to plan, and the iCore program, which is a program that helps in customer discovery and that was rolled out initially by the National Science Foundation in the US, but has been duplicated by a number of different funding agencies. We also direct funding towards our startup. Our main mechanism to do so is called the Innovation Gap Fund. We invest about $50,000 or so in our pre-companies. We don't expect any equity because the companies have not been formed, but we really can do that in a way that is effective. We also support what we call commercialization fellows, who are postdocs that will be working on a particular innovation. And we have a number of different mechanisms to support these activities as well. We do not work in a vacuum. The University of Washington is a very big place, and we have fantastic centers and programs in various departments that I am not going to describe, but that really help us to drive this innovation forward. So crossing the critical radius is something that we pay a lot of attention to. And how do we nurture this fragile early technology? How do we move them forward from this idea or this original invention to true innovation and a company? So how do we help them with the timing? How do we help them find the right execution team, which is important in some areas of innovation development? Again, we have a number of programs to do so. We help them with customer discovery training. We provide some funding, as I mentioned before. We help them write SBIR and STTR proposal, which are grants to a number of different US agencies that pair up a lab, typically that has created innovation, with a startup that will be working on that innovation and trying to commercialize it. We help our pre-companies and our innovators with IP landscape analysis and some degree of market analysis. But what really makes a difference is connection, introduction, and mentor matching, because these guys need help, and the mentor that we can provide through our network can really make a difference, because these are highly experienced individuals that can actually advise a company early on to take the right steps. To navigate the energy landscape, we really help companies address dependency related to early stage incubation, to networking once again, to acceleration, and to access to capital and funding. So we have 2 incubators that cover a number of different sectors. At local incubators include life sciences, including synthetic biology, biotechnology, med technology, and more recently, we opened a hardware incubator, which has a 5G sandbox where people can start thinking about using the high connectivity that 5G provides to think about different application or different ways of delivering their products. We also have a more traditional co-working space where some company can work on software, information technology, blockchain, and fintech, which we called at Startup Hall. We recently brought a few additional missing pieces to our ecosystem. The first one is Creative Destruction Lab, which is an outfit out of Toronto that has been franchised in a number of different places. And the CDL site in Seattle focuses on computational health. So whatever intersects, if you want, contextualized machine learning or other type of computation with health technology, ranging broadly from therapeutics to synthetic biology to medtech. We also are in the process of launching a new fund called PAC Ventures that will be funded by UW alumni and that will be investing in companies funded by UW alumni, including startups that are out of commotion. And we have a program called the Pitch, where we actually match a number of angel and venture investors with our startup so that they have a chance to actually present their ideas and seek perhaps seed funding or perhaps Series A funding with these different firms. Now, I wanted to point out that the recent success have been amazing. We've seen about $800 million per year of economic activity over the past 2 to 3 years. And let me point out a couple of startup right here. One of them is Lyell Immunopharma that raised over $600 million over a year in 2019. And another success story, which is PVP Biologics, which is an outgrowth of the Institute for Protein Design that was acquired by Takeda Pharmaceuticals in $330 million in payments and milestone last February. So what I want to do here is at this point to give you 3 stories for our spin-offs. And because we're talking about synthetic biology and the bioeconomy, I picked 3 relevant spin-offs that deal with this field. The first one is Parse Biosciences. So Parse was formed out of technology developed by Professor Georg Selig at the University of Washington Electrical and Computer Engineering Department. And 2 of his trainees and postdoc, Alex Rosenberg and Charlie Rocco, realized that they could use a combinatorial cDNA barcoding technology that, when combined with successive dilution, allowed them to do single-cell transcriptomic analysis. And that was a revelation, and that was a way to actually access information on the single-cell level. So they decided to form a company that was called originally Split Biosciences, and now is called Parse Biosciences, in 2018. And the way that commotion helped them was first to help them formulate their IP strategy. And it was pretty traditional as far as Parse was concerned. You know, we went for normal PCT and direct to US patents, but we gave them a boost. And the boost came into the form of $50,000 Commotion Innovation Gap Fund that helped them, you know, think about what they were going to be doing. We provided a commercialization fellowship, which is essentially a postdoc fellowship, so that they could continue working on the project. We gave them some money so that they could develop their beta kit. And we allowed them to meet with angels and investors and connecting them with them to give them a further boost. And this worked well. In October 18, Parse raised $1.2 million in seed, and in January 21 of this year, they raised $7 million in Series A. So they're off to a great start. We're very happy, and we look forward to what they're going to accomplish. The second story is that of OldPep. OldPep was founded by Professor Valerie Daggett, out of the University of Washington Department of Bioengineering. And Professor Daggett has been working for a very long time on amyloid diseases, including Alzheimer's and Parkinson's diseases. And what she discovered over the course of her career was a new protein structure that is named the alpha sheet, and that you can see represented right here. And she realized that the structure was associated with the early step in the formation of amyloid plaque and with the toxicity of amyloid oligomers. Valerie kept working on the topic, and she further was able to identify some complementary alternating peptide that could neutralize the toxic effect. So at this point, she had in her hands a way to diagnose potentially early on Alzheimer's disease, but also a potential cure for Alzheimer's disease. So she founded OldPep based on these alternating peptide names. out of the University of Washington in 2019. And the IP strategy was a little bit different right here. What we decided to do, and what Valerie really wanted to do, was to protect the structure rather than the composition, because this alpha sheet was unique and the structure that it adopted was fairly flexible. So we had to help her convince the US Patent Office that it was possible to protect such a structure rather than simply a composition of matter. We gave her a boost by awarding to her team a Commotion Innovation Gap Fund that allowed her to move the innovation forward. She participated along with her team with an idea to plan a workshop, and we incubated her company early on at Commotion Labs, because this is what we want to do. We want to give these early startups a chance to be successful by incubating this early innovation and then help them fly from their own wings when they're ready to do so. And the raises were actually remarkable. In August 20, OldPep raised 2.1 million in seed capital, and in January 21, just like in the case of Parse, raised $23.1 million. So these numbers are quite encouraging, and you can see that there is a significant amount of money that is going in this field, although there has been a lot of failed therapeutics in the field of Alzheimer's disease. So this is really, really a new technology. And finally, I wanted to point out the Icosavax story. Icosavax was created by Professor Neil King and Professor David Baker out of biochemistry at the University of Washington. And this was really the outgrowth of a thought process, which was, how can we design from scratch some protein that are going to assemble into cages a very, very precise dimension. And you can see here David and Neil that are holding some of these molecular models of some of these cages right here. And this icosahedron essentially were produced by Neil and David, and they showed by using a number of characterization techniques that they were assembling just as predicted. And this was the perfect platform to display viral code proteins in a virus-like context. So this approach has been taken by manufacturer of vaccine-like particles. But in this case, there was enormous flexibility because the self-assembling component was designed. And so there was a possibility to go after a number of different diseases that were causing some problems, such as respiratory syncytial virus and human metapneumovirus. And as the SARS-CoV-2 and the COVID-19 pandemic exploded, to go after SARS-CoV-2. So Icosavax was spun out of the UW in 2017 to really address the possibility that protein cage design could provide. And the IP strategy was fairly conventional, except that we had to grant the company 3 separate licenses to cover different classes of products. The boost was to give them a dub pitch invitation so they could pitch their innovation to a number of angel and VCs. And the raises followed, 3.5 million seed in December 17, followed by a $51 million Series A in October 19, and a $100 million Series B in April 21. And one of the things that we're very proud of is that Icosavax exited in July of this year with a $180 million IPO on the NASDAQ. So this is the type of things that we can achieve through careful chaperoning of our innovators along their innovation journey. We're very happy with the outcome, and we will continue to produce successful startups. So we have a vibrant ecosystem, and I am glad to share some of these information with you. And with this, I will close my presentation and thank all of you for listening.
Hello, I'm Geoff Baldwin from Imperial College, and first of all I would like to thank you for the opportunity to speak with you and be a part of this meeting. I'm only sorry that I can't be there in person, but I do hope at some point in the future that things might change and we get to meet together. So I want to, during this talk, give you some sense of the development of synthetic biology as a discipline at Imperial College as well as across the UK, and as an academic discipline, but also at that interface with innovation and entrepreneurship. So really, a bit of history. Synthetic biology started at Imperial College. We can give it a pretty precise date. It really was the first iGEM team that took part from Imperial College back in 2006. The competition at this stage was a few years old, but it was still pretty small with not that many teams. And our first team came runners-up in that competition, which was an amazing result. But the bigger result was that this really acted as a beacon to attract the brightest and the best students. And it really unleashed a wave of creativity and imagination from the students. And we had teams that took part every year for the first few years, and then we've been a little more sporadic since then, but certainly over 10 teams from Imperial have taken part in iGEM over the years. It's been a fantastic opportunity for the students that have taken part. But really back in those early days, it was quite remarkable in how it bootstrapped the interest and the capabilities in synthetic biology. And it really became a sort of bottom-up emergence of the field rather than a top-down something sort of led by the academics. It needed that coordination and leadership, but really it was the students that brought it through. So with that, we developed what at the time was the first undergraduate module in synthetic biology in 2008, and this was running, it's still pretty innovative today, it still runs as a joint course between the Department of Life Sciences and the Department of Bioengineering, something that's pretty hard to pull off and something that's still, as far as I'm aware, pretty unique in college. And we then followed that up just a year later with the first masters in synthetic biology, systems and synthetic biology. And again, there's now many master's courses across the UK covering this space. So following on from that, interest started to develop in this space and awareness started to grow from the funding bodies. And the first UK-funded Centre for Synthetic Biology was founded at Imperial College in 2009. That had 5 years of funding to run. And that really marked a sea change because that sort of enabled us to recruit a number of academics into the college and to really get some critical mass in the space of that very important and very nascent time for the field. Following on from that, then across the UK, this interest and awareness really began to take hold. And so in 2012, the UK government founded the Synthetic Biology Leadership Council, which has been really very influential and an important interface between the government science minister and UK funding councils. And that led also through to the investment for growth programme in synthetic biology, with over £100 million being invested over 5 years, that has led to a further 6 synthetic biology research centres across the UK, DNA foundries, a centre for doctoral training, and at that time as well a synthetic biology seed fund that was sort of managed by commercial interests in order to try and bring up the emergent startup scene in that space as well. This followed on in 2013 with the founding of Sydney City as the National Centre for Industrial Translation in Synthetic Biology based at Imperial College, and that was really aimed at trying to nurture this ecosystem around startup and translation. Synthetic biology as an applied discipline, it really belongs in that translational space, and pulling projects through into translation and companies is a key aspect of development in this space. There's been some important documents published along the way as well. So the Biodesign for the Bioeconomy Roadmap published by the UK government. This came out of the Synthetic Biology Leadership Council. These have been really very important and influential in terms of creating funding policies for research councils to follow, and really mapping out how we see this field developing as we go forward. So then in 2017, we relaunched the centre here at Imperial College under the leadership of myself, my colleague Guy Bart-Stan, who's in bioengineering. And then we also had Steve Chambers join us as a Royal Society entrepreneur in residence. And then we successfully founded a new doctoral training centre in biodesign engineering across Imperial, UCL and Manchester, which has now been running since 2019. So that gives you kind of a very brief history of synthetic biology at Imperial College, but also kind of across the UK. So that wider development piece and engagement has been really critically important, and the leadership, Paul Fremont and Dick Kitney at Imperial College, have played a very important role in terms of working with government and funders in order to increase the awareness and really create the opportunities that led to that Synthetic Biology Investment for Growth programme back in 2012. Notably, we are at the end of that investment phase, and this does leave actually the UK with a very important question of how we take that forward, and that's something I'll come back to in a minute. So the Centre for Synthetic Biology at Imperial College now encompasses a large number of groups across a really wide spectrum of activity, from microbial control engineering, bottom-up protobiology, and also mammalian applications. It really is a centre of world-leading research that has got a huge number of really fantastic publications coming out of it. So there's no question that we don't have, some of the best synthetic biology research going on in the world here at Imperial College. The doctoral training centre that I mentioned has also been very important. That doctoral training centre started in 2019 with a £7 million investment for the UK EPSRC, the Engineering and Physical Sciences Research Council. That funds up to 40 PhD students across 5 cohorts of intake, and we are committed to actually training 60 students because we have to leverage the money that we get from the government through industrial interactions and institutional support, so that we train more students than they directly give us money for. And when we founded this, we actually reached out to industrial partners to ask them what were their key requirements in terms of industrial training base that's required to develop synthetic biology. And this quote from Marcus Gechater at Synthase really captured that very well. And what he told us was what the synthetic biology industry needs in terms of PhDs is lab competent coders and code competent biologists with a firm grasp of entrepreneurship and other business development skills. And that kind of became the foundational statement for our doctoral training centre, and it still is our kind of mission statement, if you like, is to deliver that, because CDTs have a key role in training students who are going to go into industry, into entrepreneurship. They're not all going to become professors at academic institutions. Only something like 1% of PhDs ever do that. The rest of them do other useful things in society, like work in industry that make good products. So across the 3 institutions, we have something like 56 academic groups that contribute to this and who apply for projects, and this covers a really wide diversity of disciplines across the full gamut of synthetic biology activity. So you can see from that we really have a fantastic ecosystem here at Imperial College, right? So we have the academic center for synthetic biology, which leads on research, education, and discovery-based activity. We interface with Symbicity, who really are building that ecosystem around translation, industrialization, and entrepreneurship. And then there's also the London DNA Foundry, the Biofoundry, which was funded by the UK government as well, which again, they work with industrial partners, they work with academics, they work across the piece in order to provide some really fantastic infrastructure. And you can see the idea here, the arrow, is that somehow we have a translation pipeline. And we do all work with industrial partners, so individual academic groups work with industrial partners, the Foundry works with industrial partners, and Symbicity works to create that ecosystem with industry. And the hope, I suppose, is that somehow leads to translation of ideas into the real world. As an example of one that has actually directly led to a company and development in the real world, LabGenius, a startup which was incorporated in 2012, they're now working on AI-driven evolution engine for discovery of high-value proteins. So James Field is the CEO of that company. He's a fantastically talented guy. I was fortunate enough to have him doing a PhD in my lab in synthetic biology. He came through, as it were, the classic pipeline of Imperial College, right? So he did his undergrad here. He was a member of one of our iGEM teams. He went on to do our master's course. Whilst he was doing his PhD, he was a supervisor to another iGEM team. He took part in a number of the Symbicity training programs post his degree and got some proof of concept funding from Symbicity that led to a Blue Skies Award. He was a LEAP Fellow in 2016 and has won sort of some early Innovate UK and DSTL awards that really helped kind of contribute to the development of the founding of the company during those very precarious early days. And because James has a kind of a real vision that he's built throughout his work and his entrepreneurship, they successfully had a consortium VC investment, which has now led on to a Series A funding. And James kind of now gets around all over the place. He's becoming quite well known, shall we say, in the synthetic biology startup space. So that's a fantastic example of how the pipeline has really directly led to the founding of a company. And actually in the early days, of that company, a number of people who kind of graduated out of my lab that James knew also went to work in that company. So there was several people through that pipeline contributed to the success of LabGenius. So across the UK space, that's not an isolated example. There have been quite a large number of startups and larger companies getting involved in the synthetic biology space. This comes from a really excellent review written by Steve Chambers, Synthetic Biology Startup Survey. So really that looks like a very rosy picture, right, that I've painted. And to a large extent it is. We have a fantastic ecosystem here at Imperial College. We have amazing research. I have amazing colleagues that I get to work with every day. I have fantastically talented students that I get to work with every day. It's brilliant. But nonetheless, that translation piece, there are success stories, but it's still really hard. And so through the second half of this talk, I want to switch gears a little bit and talk about that space and perhaps to think about how we can better develop that space and increase the opportunities for success. So how do we accelerate innovation? And the idea that innovation is the product of both invention and commercialization is not my idea. This comes from Bill Odette and Ed Roberts at MIT. An invention, consists of basic research and then application of that to a problem. Whereas once you get into commercialization, you're then into a process where you need to develop a product. You need something that is of value to someone, that solves someone's problem. And there's a process around that you can do around customer discovery and customer validation, which is really the Steve Blank Lean Launchpad basis, which kind of underpins a lot of training programs around entrepreneurship now that I'm not really going to get into today. But how do we bridge this innovation gap, right? So we have basic research, and I don't know if you're familiar with the concept of the technology readiness level scale, but at 1 or 2 we have basic research, and at 9 we have a product in the marketplace. So the typical research funding that academics apply for is addressing those very low TRL level activities, which is about discovery research. Industry will invest in products that they can bring to market, and so that's obviously of a much higher TRL level, and typically takes more money as well. So that leaves us somehow with a funding gap in the middle. The overlap of these 2 triangles, to me that looks about right in terms of actually getting money to do translation is very hard, and typically, I don't know, research councils here have these sort of small schemes where they sort of, push the money out to institutions to do translation, and they come up with schemes where you can apply for 50k to translate your research. And of course, the reality is that that doesn't fill this gap, right? I mean, how do you, if you really want to bring a product to market, how do you cross that Rubicon to go from research into commercial development? So really the classic way that things get to market follows the waterfall development model. You start with basic research, you then move on to applied research, and from that you move into product development. And this is really the sort of post-war paradigm of industrial development that has been prevalent throughout the Western world for the last few decades, and it's proven pretty good at getting products to market, right? You have a focus at the end of product development, but the limitations of this approach are that it is by definition one way, and hence it's not good for discovery. It doesn't address discovery needs in basic research. And certainly in the UK, some of the industrial funding is structured purely on this vision of development, right? They won't fund you to do any research activity. It's all about product development and getting a product developed. And that's fine as far as it goes, but it's never going to get you anything really exciting. It's probably not going to create, the next really amazing, transformative company. So is there another way of doing this? Really, if we go back to the drivers here, if you think about the importance of those big questions, the what-if questions, how would you shape our world? And really, what problem is it that you're trying to solve? So if you can define a problem that matters, so people working on it care about it being successful, and there's a really good reason to do it, and it might seem like an impossible ask. So, this brings us to an alternative way of tackling some of the really challenging big questions that would be really transformative. Okay, so, this requires the same 3 facets: basic research, application-oriented research, and product development. But by creating a cycle here, we are linking basic research to product development. So we're acknowledging that we have a gap, right? A capability gap there's something that we want to do, but we don't yet have the capability to do it. And this requires basic research in order to address that capability gap. But by creating a link, a communication between basic research and product development, we're now using our creativity of research to really more tightly frame what we're doing to actually address a real world issue, right? We really want to see, can we address the capability gap that we have in order to answer our big question. And if we get an answer to that scientific problem, then we can move on to the application-oriented research, which then leads to the product development. So, as an illustration of this, take the big question, which has been around for quite a long time, the idea of gene editing. If we could edit genes, we would have the potential to solve and cure hereditary diseases. So that big question had a capability gap because we didn't have a good technology for editing genes. Basic research led us to discovery of CRISPR. So this was completely, naive research. And again, this demonstrates the importance of an active research base that's perhaps not directed at applications. But once we had the answer to that scientific problem, now you can begin applied oriented research, and you can begin to think about what that product is going to look like. The early days when you don't have the capability, you don't really know what that product's going to look like. So this approach is used out there. It's notably used by DARPA. So DARPA really are one of the most successful organizations for bringing through really transformative technologies to market. And the application of this process is the foundation of how they do that. I would also argue that innovative companies, so companies like LabGenius, also apply this type of methodology in their day-to-day organizational structure and way of working, right? So it's a really good way of connecting discovery, sorry, I shouldn't touch my mouse, of connecting discovery-based work to application-based work. So how do you actually choose what those big ideas are, right? And so DARPA, to use them as an example, have some criteria. So what they want to know is, what is the capability that we need to address a big question? And are there existence proof points in the science that kind of make it look like that's an achievable goal? So, from there you can ask what resources are required to get demonstration at convincing scale. Is that feasible? And can you think about getting there within a 3 to 5 year program? If either of those aren't successful or accessible, then it's not likely to work. And these things act as a really strong filter in terms of that ideas pipeline of which ideas you can really apply this methodology to. The other thing that they do is put that restricted time box around it. Academics are kind of great for doing exploratory, non-directed discovery. But if you have a goal, the choice of your path to that goal is really critical. And focus on that is really key, that you avoid those scientifically interesting and curious maybe, but ultimately dead-end routes that are never going to get you actually to what your defined criteria is. And the other aspect is the problem needs to be at a big enough scale, that it requires interdisciplinary working, and that no one can get there on their own, right? It requires people to pull together to kind of really create new ideas and new opportunities. So what does that require for success? Obviously, it needs money, right? And probably quite a lot of it. And it's important that that is going to give you adequate resources for the job, because if it doesn't, that's an obvious failure mode for the project. The other thing it needs is people, right? It's not all about money. You need really good people. And this is where, some of the broader questions around having a pool of talent across basic research, across applied research, across commercialization, involvement of VCs and funding agencies. And this is where we see the success where we have specialist clusters. If you look across the world, you can see tech clusters where specific technologies really take off in a geographic location. And that's because you develop the talent pool required for that. So, here the wider ecosystem of universities, of training, has a really important role to play in building that talent pool. And of course, those people need to be rewarded in an appropriate way. But again, that reward isn't necessarily all about money. If the people really believe in the project and what they're trying to do, then that's hugely important overall for success as well. And the final thing really is that absolute focus on the goal, right? So those people need to have an interaction with and a commitment to, the program, the big idea. And at the center of this, you need someone who's going to lead that activity, right? You need someone who's going to be personally responsible for pulling this through. And because there's so many moving parts here, that's going to require quite an agile coordination approach in order that you can get different people from different disciplines all pulling in the same direction. And again, I would argue that this applies to numerous scales of working. This is applicable to a startup company. It's applicable to an individual research project running in a lab. It's applicable also to a funding agency, right? If you can define your goal, you then have to muster your resources and create the structures that allow that to become successful. So thinking about, the shape of the ecosystem is really tremendously important. And I guess what I'm also leading to here is to kind of say that just training people and throwing money at them and hoping that some of them are like James Field and that they will pull it through for you and you can point at a success, that only ever gets you so far. I think really to create a truly fantastic cluster of innovation, you need to start to put structures in place. If you're going to provide funding, you need to think about the nature of that funding, what you're asking people to do with that funding. And if you really want to drive innovation, you need creative ways of working. So sort of imposing a waterfall-based model on a research group or an early startup company is not a route to success for that company. And the reason I say that is because some of the funding that I've seen applied and had myself in that space has attempted to do just that, and it doesn't fit the mode of working. So we need to recognize that there are different modes of working, there are different ways of working, and that we need to create structures and opportunities for that. I think in the UK, we have many of these things in place, but we don't do all of them as well as we could. So that comment about the appropriate structures of funding and organisations, I think, we are seeing things change a little bit in the UK. I don't think they're changing fast enough. One of our biggest problems, I think, in the UK is this bag of cash question, because if we think about synthetic biology, yes, we had 100 million investment back in 2012, but that was 8 years ago. That's now run out. I think phase 2 or phase 3 of synthetic biology is going to take more than that. actually. And I think there's a real systemic risk for the UK that we will not have the resources required to do the job for phase 3 of synthetic biology. I think in China and the US, there are huge opportunities there in terms of the resources that are going into SynBio in China are pretty staggering compared to the UK. But it's not just about money. You know, there is that real question of how you structure that and how you create the opportunities and the structures around those opportunities in order that you really try and facilitate the development of good ideas, the development of innovation, and the development of commercialization. And with that, I will leave it there for now, and I very much look forward to joining you in the discussion group later. Thank you.
Well, thank you. And thanks to the organizer and Professor Cheuh for inviting me. It's been a really exciting morning. So I changed my slide in the very last minute just to answer to Harvey's talk this morning. And we have heard some very exciting news from Washington and also UK. So I actually have changed the title of my talk. Can we have this slide? And instead of going into UK and US or even China and GBA, I decided to talk a little bit about what we're doing here in Hong Kong and how could Hong Kong contribute in terms of synthetic biology back to GBA and China. GBA stands for Greater Bay Area, just for the record of it. As some of you may know, I'm not a scientist anymore. I have 0 publications in the last 2 years at least. So what am I going to do here before Cheuh would query how and what I'm going to present? So I'll give you a little bit of introduction of myself. I rarely do this in talks, but I decided to do it this time. This is my PhD supervisor, Dr. Xiang Lin. And we met in Shanghai here about 12 years ago. The first question she asked me was, are you still working on Saturday and Sundays? And then embarrassingly, I said, only on Saturdays. And she was not too happy about it. At that time, I was head of Roche for the entire Asia. That's because she came out of this guy, Irwin Chagav. For some of you who know, this is the person who matched A to T, C to G, adenine to thymine, and cytosine to guanine. But he did not get the Nobel Prize. The Nobel Prize went to this guy. And we invited James over, actually along with UST some time ago. But what really shaped my career in molecular biology, at least, was this. At that time, the US Cold Spring Harbor decided to train the molecular biologists in Asia for the first time. But they could not enter into China, so they came back out to Hong Kong and actually did the training there. This was the first training outside of the US, and these are the first, or these were the first batch of groups who actually tasted molecular biology cloning at that time. The manual was not even ready. It was given to us at the end of this training. And this guy on the back here, for those on site, you'll be able to see the tall guy in white hair. He is Douglas Hanahan. For those who do cloning, you use E. coli. DH1, DH, blah, 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 DH7. DH stands for Douglas Hanahan. He was a postdoc at Harvard at that time and the youngest. He's now in Switzerland. There are also somebody in the middle who became the chief scientist for South Korea and the head of the Chinese Academy of Sciences in Taiwan, who's somebody at the back, and I'm the one on the left. It was with this course that got me into molecular biology and cloning, and my PhD. But after my PhD, or during my PhD, somebody came from UCSF, and he was Harold Varmus. We named it Varmus, but he told us you should call me Vamus. Anyway, he spent 6 months here in Hong Kong. Nobody knew who he was. He was trying to put a relation between RAS gene and liver cancer. And that's why, because of working on liver cancer, he came to Hong Kong. So it was great knowing him and his lab. And in actual fact, we got the first set of oncogenes from him. And that started some of the cancer biology research in molecular biology in Hong Kong for that time. And then after I got my PhD, they said, you know a little bit about molecular biology and DNA, but you don't know enough about cell biology. So they sent me to do cell biology. And I was glad to have a chance working in Cesar Milstein's group, and where we actually had my first commercial product out. And it was a murine protein that was sold to Boehringer Ingelheim. And then I joined the industry afterwards. And in actual fact, it was a company that was right opposite to Genzyme in Boston, to Harvey's company. But there were two guys that we have to report on a regular basis, which we have learned so much. That is Philip Sharp and Waterfield. And Craig Mello came in as a very nice story too. Somebody from Wall Street wanted to talk to him, asked me to join. This was before he got his Nobel Prize. He actually made us coffee in his lab. And I joked that this was the best coffee I ever had. After 2 months afterwards, he got his Nobel. But the real picture I want to show is this one. The father of my supervisor is actually Lin Yutang. For those in Asia, you probably know him. For those, if you don't, you don't. But what I was fascinated was he was actually an inventor of Chinese typewriter. So there is something in the genes of invention. So today, I think I have about 16 patents today. Half of those have been reduced to practice. For this talk, which caught me a little bit because I have not been involved in synthetic biology recently. So I went back to the literature, and these are some of the references that I've gone through and pulled stuff here. And actually, there was one report from the UK that was pretty good. In any case, I don't think I need to go into define what synthetic biology would be, because it's just preaching to the experts. But basically, it has a pretty long history now, over 20 years. And at least for that course to now, it's almost 40 years. It actually deviates from pure biology or pure engineering, whereas it is going to be using interdisciplinary efforts to come up with new models and products that would hopefully do human good. And basically, there are lots of applications from a wide range of products, from medicines to agriculture to even cosmetics and host stuff. And this is a chart summarizing what has gone through in molecular biology. And those folks that I have shown you in the slide probably will be squished to the left column, whereas the rest would start from the turn of the century, from sequencing of the human genome. I think synthetic biology really took off. There are about 2 or 3 really key important inventions that got synthetic biology going. I think it's from the human genome, and then it was the Gibson assembly, which is piecing DNA pieces together to form a longer strand. It sounds easy, sounds simple, but actually that's the basics for building synthetic biology. And then clearly, CRISPR-Cas9, this gene editing technology, would definitely be another piece of innovative and going to have a lot of groundbreaking inventions coming around. Clearly, we have done quite a bit in synthesizing DNA, and we are actually building artificial bacteria ourselves. What would synthetic biology include? I would only look into 3 types, and that is products, technology, and applications. And this is just a different way of skinning a synthetic biology definition. You can use a whole range of other approaches. Definitely, it would include a lot of disciplines from bioinformatics to genetic engineering, etc. Here are a couple of examples in terms of enabled manufacturing. That is how synthetic biology would enable manufacturing. This particular company called Impossible Foods, what they did was they just add heme back to some of the artificial foods or meat so that it now tastes and actually smell like real meat. It's just a simple thing. And then both threads actually was using a spider silk protein and use that to be produced into a textile raw material that would be used subsequently in our everyday products. The Pfizer BioNTech or Moderna vaccines using mRNA as the bait to produce the antigens for battling COVID-19. That's probably one of the best examples recently for synthetic biology in the field of medicine or vaccines. We tried to bring Moderna into Hong Kong, as some of you know. That was 5 years ago, way before COVID-19 happened. But our experts said, after SARS, Hong Kong had no infections. Therefore, we don't need it. So there it goes. Speaking of readiness, anyway, Pivot Bio has another great technology that put nitrogen fixing pathways into some other means or into some other organisms so that they would replace the use of ammonia-based fertilizers. Those are things that we learned from basic biology all the time. It's just how you put all these things back into applications. Of course, as some of the speakers earlier have mentioned, the investment into synthetic biology has been pretty exciting. And you can see this amount of investments going into synthetic biology startups has been going up like that. But we still expect a lot more to come by. In the US, they have a different kind of definition. This came out of a report coming out of National Academy of Sciences, National Academy of Engineering, and National Academy of Medicine as a joint report on synthetic biology and bioeconomy. And they are trying to define several sectors that they are going to focus on, which is biomedical, agriculture, and biomaterials. All others they will put into miscellaneous. We call it miscellaneous, but they call it toolkits and devices. And they gave lots of examples on each of them. So depending on how you define all these areas, it will actually help the government or policymakers to define their policies when you want to develop this particular field. Now, what caught their eye was because when they looked into the biotechnology revenue, this was back in 2017, they realized that it was already a whopping 388 billion revenue, or 2% of GDP. And today, it's already more than double that number for those who are interested in economy. But mind you, R&D services amount to all the possibilities. It's only occupying a very small percentage of the economy. That is, these are contract research given to either industry or the academics. Majority of the revenue is still coming from biochemicals. Now, this was coming from, I believe, McKinsey. They predicted that in 20 to 20 years' time, the bioeconomy would grow into 2 to 4 trillion. These are all in US dollars, size market. And this is also because when we're interested in synthetic biology, it is not just because of the economy, that is the numbers and the dollars. Obviously, it's attractive, but it's also because of its impact on health, but also on environmental sustainability, when the UN has already set out its goal to really control CO2 emission, etc. And that is, a lot of this synthetic biology technology can be applied to improve waste management, reduce land and water use, reduce carbon emissions, and more importantly, as a biologist, to reduce the loss of biodiversity. That is, even things that were extinct, maybe one day we could recover them from a genetic standpoint. This came out of the McKinsey report, and they consider there will be 4 areas of bioinnovations, from biomolecules, that is some of the chemicals, to biosystems, and then bio-machine interface and biocomputing. The biocomputing, just now, even in Geoff's talk, I think they're already talking about cell and actually tissue organ type of computing. But the important thing is the economy and the contribution of all these biologies will be significant compared to all other type of engineering type of development. And the fact that synthetic biology has been successful, other than the effort from all the scientists, was because of 2 factors. The cost of sequencing the human genome has been going down, and the speed of sequencing the human genome has been going up tremendously. Makes it an efficient project, efficient process, and cost effective. There are several 2 trends or application trends and/or application trends coming out of synthetic biology. We can either focus on gene drive, coming out with animals that can do something else, like what Stanford has been doing, putting mosquitoes out to get the Ebola virus out, or CRISPR tools doing gene editing, bioinformatics to analyze the genome, cases development, trying to know what is needed as a fundamental structures, and then obviously DNA synthesis. And that could put into applications like human genome editing, plant genome editing, biosensors, metabolic engineering, etc. I'm not going to go into all the finer details of those application trends, but you can imagine there will be specific applications that we can call upon under each category. But I would focus on one thing, and that is the synthetic cells. In actual fact, this morning, Harvey actually talked about that when he was mentioning the RBC experiments that he and his colleagues have been working on. And these synthetic cells are not liposomes, not bacteria, not eukaryotes, but they have everything inside except probably the reproductive capability. And these are structures that we should pay attention to, as this, I believe, will be the next wave of inventions. Liposomes have been successful. Looking at the Moderna and Pfizer story in the vaccine. But Bob Langer's company, Sigilon, just had their liposome as a delivery mechanism for gene therapy on clinical hold because of safety issues. This just gives you a detailed look of what a synthetic cell could possess. It is whatever you can imagine in terms of biochemical pathways or surface protein receptors that you want to put in, you will be able to do so. Just now we heard of the term biofoundries. They are actually experimental grounds where scientists would be able to learn, design, build, and test all in one place. It is not built and then to test. It is changing all the time. And one of the key features of a biofoundry is that it is almost fully automated, or it relies heavily on automation. Clearly, when we're talking about synthetic biology, synthetic cells, there are two issues. Scale up, it's still a big problem. How to bring it from laboratory scale into a manufacturing scale. And safety of all these new entities, just like some of the synthetic cells that I've mentioned. In China, it's going to be successful, as everybody has predicted, because they are investing a lot, and there are some very successful institutes coming out of even Shenzhen. And these are examples from the Center of Excellence in Molecular Plant Sciences, and they just had 2 publications in Cell in October, one on the 14th, one on the 28th, specifying very important features of understanding immune responses in plants or some of the phosphate starvation responses. I think it was also in wheat. So these are important discoveries that would allow them to really focus on agriculture. And because the US is so aware of this grand economy of 2 to 4 trillion, so they actually published a paper to safeguard the bioeconomy, how to make sure that they would be in a leadership position. I won't go into all the details, but basically, they want to establish a government-wide strategic coordinating body to drive the development and to safeguard their interests. And obviously, asking for more investment and more collaborations. Also, Johns Hopkins published this one, and that is to analyze if US and China are in this strange, sensitive political situation, how would that affect actually synthetic biology? And they call it decoupling. How would it become when China and US decouple? And the answer is that China must develop her own INT. Right now, US can afford to take the hit for a temporary, painful period, but then they will be fine. But China may not be as good. So China would need really to go into innovation, etc. Just like all new sciences, synthetic biology would also have one sore spot other than what I've mentioned, scaling up, safety. The other thing is, what are the possible risks? And usually it is data privacy, equity, credibility, and whatever that is unknown. And the unknown is definitely what was being held in Hong Kong about 3 years ago on a gene editing baby. And this is also what Hong Kong can provide as a neutral ground for international experts to gather, discuss, and also to come up with solutions. This is our Vice Chancellor here at UST. And also we're getting quite a bit of funding from the private sector to start synthetic biology. There are other colleagues in other groups, and actually they are from Harvey's lab at City Poly University doing exactly what he was talking about. At the Chinese University of Hong Kong, where I work for now, what we're trying to do is to start a blockchain platform for Chinese medicine, traditional Chinese medicine. We believe that with all these, eventually it would spell or it would push synthetic biology into traditional Chinese medicine, herbs, that would make it really unique for China, Hong Kong, and the Greater Bay Area. So this is our Vice Chancellor from CUHK and Professor Lam Hon Ming. He's known as an expert in soybean genome. And therefore, what he did was, over the years, he tried to identify new varieties of soybean and the genes related to salt tolerance, heat stress, plant architecture, nitrogen fixation, color traits, etc. That is to get better soybeans. And eventually he got them. But more importantly, he planted all these in a province called Gansu. It's a really poor province. As you can see from the landscape in the slide, it's almost like nothing. But now he would be able to make use of this fairly poor condition to come up with soybeans, which is a key component of the Chinese food. And more importantly, when he has been identifying all these new varieties, they're reducing the use of nitrogen fertilizers, reduce carbon dioxide release, indirectly reduce PM 2.5, that is getting better, cleaner air, and helping the farmers to get better income, which is indirectly helping the bioeconomy. He himself did not get 1 cent out of all of his patents and inventions. He donated everything to China. And finally, just to round up, Hong Kong can be a major contributor to synthetic biology. We know what we can do. We're very good at international network. We can do talent flow, finance collaborations, IP trading, etc. But what we can add, actually it's IP registration, litigation, and arbitration. Okay? The other thing is we should really help China to build an APCIEE-like cell bank. This is not happening in China. It will offer central registry, storage, and distribution of materials, coming with central standards, policy, and ethics. And like APCIEE now, they can actually service the government and service other industries. I don't need to go into that. What China has, they have 6 isolated cell banks right now, but not coordinated and not representative. So that is not going to help synthetic biology in the long run. So we really propose to the Chinese government, hopefully, to set up such a bank. And this slide was added just now in response to Harvey's. We have written a proposal and submitted to the government to form a center of rare diseases. We've got some funding already, but we really want the government to be part of it. And that would really help all of the rare disease patients, not just the 20 million in China, but actually all of the rare disease patients in the world. That's the vision. With this, I would like to end the talk and emphasize that synthetic biology would be a new science that hopefully can bring more goods to human than pure money and monetary return. Thank you.
Thank you, Professor Lee, for your sharing. Now, may we please invite Dr. Cheuh Po, Associate Professor at the Department of Biomedical Engineering in the National University of Singapore and the co-founder of Advancing PTE Limited in Singapore. The keynote lecture will be about synthetic biology in Singapore. Dr. Paul, please.
Yeah, thank you very much. Can you guys see my screen all right?
Yes.
Okay. So yeah, so very nice to be here. So first of all, I thank Prof. Cheuh and the organizer for inviting me to be part of this very interesting panel and to attend this very interesting conference. So it's my great pleasure to be here to share with you a very high-level snapshot of synthetic biology that's happening in Singapore. Now, to put things in context, as you guys know, right? So Singapore is really a small modern city country located in Southeast Asia. And we have a population around 5.8 million people. And we are really a small island, about 40 kilometers from east to west. So what I'm going to talk about today is really, within this context. So what have we been working on, in synthetic biology? But first of all, to talk about synthetic biology activity, let me first introduce the Singapore National Strategy for R&D. So the Singapore government is actually very committed to research and development. So over the years, research, innovation, and enterprise are the cornerstones of Singapore's national strategy. So the effort is actually to develop Singapore into a knowledge-based, innovation-driven economy and society. So to structure this strategy, every 5 years, the government will review and craft out a strategic plan for the next 5 years. So the most recent one is the RIE 2025 in short. Now this covers the year from 2021 to 2025. Now for this particular RIE, the Singapore government has committed 25 billion Singapore dollars over the next 5 years. So if you take a look at the RIE, essentially it's organized around 4 strategic domains, supported by 3 cross-cutting horizontals, right? So the 4 strategic technology domains are the manufacturing, human health, urban solution and sustainability, and last but not least, smart nation and digital economy. So essentially, in terms of synthetic biology, it fits into at least 3 of this, right? From manufacturing to human health and to sustainability. Now, and then with that, these 4 areas are supported by cross-disciplinary kind of horizontals. In terms of academic research, which deals more on the fundamentals, to manpower development, and last but not least, innovation and enterprise. And this is something that I'm going to focus more on. So in terms of technology development, we recognize the importance of translating technology from research to the industry. And to facilitate such translation of technology, technology consortia, which cover different areas of research, they have been established by NRF. So NRF is the our Singapore National Research Foundation, to kind of catalyze private and public partnership. So Synergy, which is the Singapore Consortium for Synthetic Biology, is one such technology consortium that's established in 2017. Now, the main goal is to really foster interaction and co-development between the industry, the universities, and the research institutes. So far the consortium has around 20 industrial members and around 8 academic members. And one of the goals, as I mentioned, is to really encourage the interaction between the academic and the industry, and helping them to co-develop technology. And to meet this goal, Synergy organize events to promote interactions, and at the same time, provide seed funding, in which the members of the consortium can apply and then carry out joint projects. So these are mainly small-scale projects, really to get things started, to get the academic to start off working together with the companies. And at this moment, over the years, we have been working on synthetic biology, and now we have a growing ecosystem and a kind of a growing research capability in synthetic biology within Singapore. And if you look at our local ecosystem, it comprises research performers from various research institutes, university and polytechnics. We also have a bunch of funding agencies, and then with industry as industrial users, right? So essentially, at the start, in like around 2015, Sin City, which is a synthetic biology research center, was established at University of Singapore, and that forms a big player in how we push, the synthetic biology research moving forward. So let me now talk a little bit more about how we go about trying to promote the private and public partnership. So through Synergy, we have actually several schemes implemented, which I will briefly cover. Now, firstly, for example, we have a national level synthetic biology R&D program, which supports the collaborative research projects, focusing on translational research. So let me talk a little bit more about this first. So what is this Singapore Synthetic Biology R&D program? Essentially, it's a nationwide integrated program, involving researchers from various universities and research institutes. The program focuses on the fundamental tools development to enable more efficient engineering of biological systems. At the same time, we're also creating a library of proprietary national strains, and at the same time to look at the strategic application and industrial relevant projects. Now, if we dive a bit deeper into this program, essentially we have 8 collaborative research projects, with researchers from NUS, NTU, and research institutes, and then coming together to achieve a common goal, right? And this common goal is set up by NRF, the SPP program, which relates to the production of high-value bioproducts, right? So to date, the 25 million Singapore dollars have been awarded to all these different projects, and a number of patents has been filed, right? So we're working to encourage industrial uptakes of this technology. And this is actually an ongoing project. It's a 5-year project, and we'll be happy to share more outcome from this program in the future. So besides the synthetic biology program, Synergy also supports early stage public private collaborative projects. This includes providing seed funding, to support academics to translate technology and know-how in collaboration with the industry. So as I mentioned, so Synergy right now has around 20 industry members, and we have about 15 collaborative research projects being carried out in various area of interest. And this area of interest spans quite a wide range. They include sustainable bioproduction relating to production of high-value chemicals in a more sustainable way. We also have interest in automations to deal with the biofoundry. How can we build our biological system at scale? At the same time, we also look at DNA as a data storage medium, just to name a few. So it pretty much covers a pretty wide range of interests, in synthetic biology. So, at the same time, I also like to mention about the biofoundry. As mentioned by the earlier speakers, in the UK, there's this DNA biofoundry. So the biofoundry established at NUS, in fact, it plays a key role in how we catalyze the public and private partnership. The biofoundry, as mentioned by the earlier speaker, is equipped with state-of-the-art automations, right? Like liquid handlers, and at the same time, we have high throughput measurements instruments. The purpose is really to enable us to engineer biology at scale. Essentially, how can we overcome the complexity in engineering of all this biological system? And then the biofoundry, which is based at Sin City, has played a key role in allowing us to engage companies. And several private and public partnership has been established through this whole use of the capabilities that we have at the biofoundry. So in addition to small scale industrial projects, the last example I have here is really the Industrial Alignment Fund that has been implemented, and to support much larger scale research collaboration with the industry. Just give one example that we have, which is an ongoing project. This is a collaboration that taps into the Industry Alignment Fund. Is the Wilmer at NUS Corporate Lab. So essentially, this corporate lab is established, with a focus to develop healthier food products and establishing sustainable production of industrial enzymes and biochemicals. So the nice thing with this corporate lab is that it's jointly funded by NRF, NUS, and Wilmer, which is the company partners. And then in this corporate lab, the researchers from both the academics and the companies, they're all co-located at the same location. And we find this to be really, really important because this really facilitates the joint development of the technology. So in fact, the academics researchers together with the company's researchers, they interact almost every day, right? And this is a really very exciting development that we have. And this is actually an ongoing project that you're looking forward to seeing more outcomes coming up from this corporate lab. Now, besides emphasizing and catalyzing public and private partnership, we feel that the manpower development is also very important to support various activities in synthetic biology research and translation, like what Geoff mentioned during his talk about the manpower and talents that is required. So over the years, we have been working on activities to promote and increase the awareness of synthetic biology. This includes us publishing a comic book, right, with our local writer about synthetic biology. And the purpose is really to reach out to our younger generation. And we also have been actively participating in iGEM since 2008. Now, this is the international student competition in synthetic biology. Now, over the years, we have teams from NUS, NTU, and Singapore Polytechnic participating. And yeah, I'm very excited to share that this year, NUS, the iGEM 2021 actually came second in the undergraduate category. So overall, we find that the iGEM is actually a very good platform to get students excited about synthetic biology. And then in fact, we have several ex-iGEMers right now continuing pursuing in synthetic biology research as a postgraduate students or eventually getting into companies like Illumina, right? Continue their sort of synthetic biology journey. So at the same time, as our synthetic biology community grows, we founded the Society for Synthetic Biology Singapore in 2019, to further promote the advancement of synthetic biology, and then at the same time to increase the awareness of synthetic biology in Singapore. This society has been organizing events to engage the synthetic biology community, particularly the students and postdocs, right, or early career junior professors. And in one of the recent events, we invite speakers to talk about the various career paths, for students and postdocs working in synthetic biology. Essentially, we want them to see, to become more aware, what sort of career opportunity, they can actually embark on when they start working on synthetic biology. At the same time, we also organize events where we get synthetic biology companies to share the ups and downs, the opportunity working with a synthetic biology company as well. So essentially, all this activity is to try to create a sort of a vibrant members of the communities in Singapore, to get people interested and continue pursuing a career, in synthetic biology. And last but not least, we also believe in collaboration with international partners, right? So Singapore Biofoundry is one of the 16 founding members of the recently established Global Biofoundry Alliance. So Geoff mentioned about DNA Biofoundry at London. They are also one of the founding members. So this Global Biofoundry Alliance is where the public biofoundries from around the world, we come together and to work to develop best practices, standards, and capabilities, for the bio-based economy. And we felt that coming together, we should have a better means, to solve a lot of challenges that we face, right? And to date, since the launch of the Global Biofoundry Alliance in 2019, we have now about 30 members from around the world, and it is still growing, right? So we believe this international collaboration among different partners, is going to help us to accelerate the development of our synthetic biology capabilities in the next few years. Now, with this, I will summarize, right? In closing, Singapore has made significant investment in synthetic biology, and we are committed to continue growing synthetic biology, together with everyone else in the world. Now, we see that synthetic biology will be instrumental, in addressing various sustainability issues in Singapore and around the world. So with this, I'll close my presentation. And then if anybody is interested to find out more, please don't hesitate to reach out to me. And I'm looking forward to the discussion panel session later. Thank you very much.
Thank you, Dr. Paul. Now, may we please invite Mr. Kenny Cheuh, Assistant Research Director and Head of Economic Development at our Hong Kong Foundation, to give us a keynote lecture on the topic Strategic Collaborations between Hong Kong and Shenzhen in Biotechnology, Capitalizing Opportunity in the Loop for Policy Innovation. Mr. Cheuh, please.
Okay, thanks for inviting me. And today we have a lot of professors and real experts in this industry. So as a think tank, my session will focus more on the public policy to see how we can help to collaborate between Hong Kong and Shenzhen. Okay, so we have the slide. So in fact, our Hong Kong Foundation has published a report in July, and it is a joint research with the other two famous think tanks in mainland China. The Shenzhen one is called the CDI, and the Beijing one is called the CCIEE. So actually, we focus on our competitive advantage areas. For example, our Hong Kong Foundation will focus on the biotech industry development and also the Hong Kong policy. While for the CDI, they will focus on the cross-border regulatory harmonization and also the Shenzhen policy. While for the CCIEE, they will focus on the central government support. So we talked to a lot of famous expert and stakeholder, and in fact, actually, Professor Daniel Lee is also one of the key advisors. So before talking about the collaboration, let me first introduce about the competitive advantage or competitive edge between Hong Kong and Shenzhen. So in Hong Kong, we have two major competitive advantage. The first one is about our strong basic research, and it can be supported by our state key laboratories, and also we have a lot of renowned professors like Dennis Low and Nancy Yip. And another competitive advantage is our financial role. So actually, Hong Kong is already the world's second largest biotech financial center, just behind the NASDAQ. And it is thanks to the Chapter 18.8, so that the pre-revenue biotech company can list in Hong Kong once they finish the phase 1 clinical trials. Well, for the Shenzhen, their competitive advantage is the high R&D spending and also the comprehensive biomedical industry training. So actually, Hong Kong and Shenzhen do have some collaboration, but just project by project. We don't have a full plan to how to collaborate each other. But lucky that we have the loop now. So the loop can try to connect Hong Kong and Shenzhen. And in my presentation later, I will talk about some recommendations to show you that how we can build the loop to the biotech industry cluster. So let me first talk about some areas for the collaborations. I know today we have a lot of experts, so I won't go into detail for this, but just want to highlight one thing that actually the whole process for the medicine is quite complicated. We have a lot of steps. So Hong Kong and Shenzhen actually can work together in each step. It doesn't mean that Hong Kong just do about the basic research and Shenzhen just do the production side. So let me take the just vaccine as an example, which is also our first area of collaborations. So Hong Kong is doing pretty well in the phase 1 trial. So, and also phase 1 trial do not require a lot of patients. Sometimes even healthy people can do so. So Hong Kong can support in the phase 1 trials. And as I mentioned, we have the Chapter 18.8. So once you finish the phase 1 trials, you can list in Hong Kong if your company is mature enough. or if your company want to develop further, actually you can find the Hong Science Park to further develop. But for the phase 2 and phase 3, actually we can work with the Shenzhen because in Shenzhen or in GBA, they have a lot of hospital and a lot of patients. So we just need to bring our clinical trials experience and standard and cooperate with the Shenzhen so that we can do the phase 2 and phase 3 together. And once you finish the phase 3 trials, there is a MMPA subaudience in the list right now. So you can just connect to the MMPA for the approval process. So you can see it is a whole process that how Hong Kong can cooperate with Shenzhen. And for the second area is about the genetic testing. However, the current challenge is that in Hong Kong, the Hong Kong company is viewed as the foreign company. So there is some negative list that's talking about if your company, your major business is talking about related to the genetic, you cannot cooperate, you cannot operate in the mainland China. So we are talking about whether it is some possibility like that if your biotech company is registered in the loop, and also the founder and the major key shareholders are the Hong Kong permanent resident with Chinese nationalities, then whether Hong Kong company can exclude from the foreign company list. And while for the another two major areas is about the advanced medical device and also the application of AI to the biotechnologies. Actually, these two areas can perfectly make use of the competitive advantage of Shenzhen and Hong Kong because Shenzhen is good at the program design and Hong Kong is good at the clinical design. And Shenzhen, they have a lot of software development talent. And Hong Kong, we have the comprehensive medical system and experience in the clinical trials. So actually, we can work each other together so that we can leverage the two cities' talent pool and also the industry advantage. Okay, for the recommendation part, as I mentioned, if we want the loop to become the biotech industry cluster, the key is to have the anchor institutes. But what is the anchor institute or anchor institutions? It can be the leading biotech company or enterprise, or we can develop the local unicorn, or we can set up some biotech mega research institute, just like the BALDWIN Institute. So for the short term, we should attract the famous leading biotech company to come to the loop. But how to attract them to come to the loop and also set up the regional headquarters in the loop? I think the key is the test and also the when. For the test, we have the corporate test and also for the R&D test. For the corporate test, actually, we can learn from the Singapore because the high-tech company with the headquarters in Singapore, actually, they can have the test exemption for the several first years. While for the R&D, some people may ask, oh, actually, Hong Kong, you have the tax reduction. You have the 200 or 300% for the R&D tax concessions. But for the biotech company, it is a little bit different because the tax reduction is only applied for the company who already have the revenue. But biotech is a high tax, so it takes years for you to the basic research to the final product and then get the profit. So what we can learn is from the Australia. Actually, they will give you the direct allowance. For example, if you invest some money in the R&D, they will just give you 30 to 40% directly. So it is good for those biotech companies because you don't have the revenue at the very beginning, but you still have some allowance. Well, for the rank, you can obviously see that the Hong Kong Science Park, our rank is really high, not just compared with our competitive city like Singapore. If you compare with our neighbor city like the Shenzhen, the gap is very huge. So we need to think about how to lower our rank or give some subsidy for the rank. And for the medium term, we need to help our local startup. How to help them? Actually, we can provide some funding so that they can escape from the value of death. If you look into the some report from the National Development and Reform Commission, actually they mentioned the government can try to set up a funding to support. So in that case, the government from the Hong Kong, Shenzhen, and even the Guangdong, they can set up the Shenzhen Hong Kong Biotech Collaborative Development Fund to help those startups to try to escape from the value of death. And for the long term, the building the mega research institute is very important. So some of you may want to know what is the mega research institute apply for. It is talking about you like to share of a substantial funding and advanced facilities. And also it will be a cross institutional collaboration and cross-disciplinary collaborations. And one of the famous example is the Broad Institute in the US, which is supported by the MIT and Harvard. So if you look back to the Hong Kong, actually we have a lot of top university. We have Hong Kong U, CUHK, and HKUST. So actually these three universities can cooperate together, and even with some university in Shenzhen, so that we can try to build a mega research institute. And there is three key principles for setting up those mega research institutes. The first one is the research is better focused on the cutting edge research, so that you can attract the cutting edge research. We have some examples like the stem cells and rare disease, so that you can attract the international top talents, because it is not just talking about the money and funding, because every city can provide a very attractive funding or money to those top people. So they are looking for is whether they can work with some top people to solve the cutting edge issues. Another principle is talking about the resolve the regional problem. For example, like the lung cancers, it is quite common in China or Southeast Asia. So we can have more experience, more patients for that areas. And the last principle is talking about these mega research institute must be operate independently from the university, so that they won't have restriction on the finance or operation restriction in the universities. And our second recommendation is talking about we need to think more comprehensive, so that the Hong Kong government need to formulate a blueprint for the biotech development. So in that case, actually, we can learn from the US and Singapore. They have the science and development office, so that they can have a blueprint about talking about the advancement, development of the biotechnology. And more importantly, they will have the funding support and talking about how we use the funding to support our blueprint. Another recommendation is that if you look into our mainland China, actually, Beijing and Shanghai is doing pretty well in the biotech industry. And you found that they have a lot of GLP labs. But if you look into Hong Kong and Shenzhen, we just have one or two in these 2 years. So we need to establish more GLP labs in the loop for the preclinical research. And even for the clinical research, I mentioned that Hong Kong is fine in doing the phase 1, but we have a witness that our approval time is really long. It takes around 6 months, which is almost 6 times for those in the Australia. So we need to figure out some way to try to cut our approval time for the phase 1 clinical trial in Hong Kong. And for the phase 2 and phase 3, as I mentioned, we need to cooperate with Shenzhen or the other cities in the GPA, so that we can have more patients, and then more patients to work on the clinical trials. And actually, we can look for the model like the Hong Kong Shenzhen Hospital. It is also talking about using the Hong Kong style management in the mainland hospital. Another challenge is that in Hong Kong, we don't know how to approve the new drugs. So the current situation is that we have a list, and in that list, we have 32 countries. And if any new drug is approved, any 2 countries in this list, then Hong Kong will automatically approve this new drug. However, if you look into the detail, you will find that China is not included in the list. And this list was not yet updated for at least 10 years. But if you look into the China, actually the MMPA, their standard has improved a lot since 2017. And it is now under the, it is one of the member of ICH. So we are talking about whether we should rethink about the list and adding China to the list so that we can also send in some positive signal for those Chinese company. And also in the loop, we are talking about whether we can build up some production line because we have some space. But some of you may think, oh, maybe the space is still not enough. So we can start with some frontier products, just like the mRNA. Their space may not be, requirement may not be very large. And other recommendation is talking about we need to set up a one-stop biotech service platform. In this platform, it can connect the Hong Kong and Shenzhen. And no matter those companies is looking for the funding support or looking for the national authority to approve some the clinical trial or anything, this platform can try to help them. And also, as I mentioned, actually the MMPA has the subsidiaries in the loop. So once you finish the clinical trials, this platform can try to help you to connect with the authority to help you to speed up the whole process. And last but not least, we know that the university, especially in Hong Kong, our basic research is very strong and it is one of our main advantage. But our technology transfer is not that good actually. So we have three recommendations on that part. For the first one, you can see that although our university system is quite similar to the one in the UK, we have the KPI for the teaching and research, but we do not have the KPI for the knowledge transfer or commercialization. But if you look into the 2020, actually the UK already published the knowledge exchange framework. So I think the Hong Kong can also consider the similar framework to set up some KPI to encourage the university to focus more on the knowledge transfer. And the second one is talking about the regulations on the professor outside work. In the current situation, the professor is only allowed to work 1 day per week. And this 1 day is already included weekend, public holiday, and annual leaves. So that means if the professor choose to work on Sunday for some outside work or cooperate with some hospital, it already used that day. So you cannot choose to do the outside work in the Monday and Friday. And the third part is talking about the talents. Actually, we have some undergraduate, we have some talents in the biotechnology, and also we have some talents in the business areas, but we seldom have talents who know the technology and business together. And actually, I found that UST is already setting up some course to try to train up some talents who know the biotechnology and business, but the size is still not enough. So I hope the university can try to teach more talents so that we will have the talent to support the whole biotech development. So that's all for my short presentation. Thank you.
Thank you, Mr. Cheuh, for your presentation. Now, may we invite Dr. Youming ZHANG, Director of Department of Engineering Biology at Shenzhen Institute of Advanced Technology and Chinese Academy of Science in Shenzhen, China, to talk about the topic synthetic biology in China, from technology development to applications. Dr. ZHANG, please.
At first, I would like to thank the organizer to give me the chance to have an introduction of synthetic biology and its application in China, especially in Shenzhen. So my presentation title is Vista of Industry. This actually is a compilation of synthetic biology industry. So as everyone knows, there's a rise of the cities and waves of technology innovations in our history. First in 1980s, in 1850s, the Glasgow has shipment manufacturing, and then in 1910s, the Detroit, USA has automobile manufacturing, and then nowadays, since this century, the San Francisco, USA has IT industry. But what's the next in the future? So we think the future will be the biotechnology and informatical technology, the compilation, actually, it's integration into together and to form a new area of the industry. Just example, what the IT and BT integration. So in this feature, you know, the structure of a protein or enzyme was previously we have to use the biotechnology tools to get the structure of the protein, but nowadays we can use the AlphaFold, the computer system, to actually to very easily get the structure, and the both actually the result from the lab and from the computer science are very similar. Actually, it's nearly the same. So actually, we can combine the IT and BT together. The other example is the lab man, and this robot can do all the experiments and replace the scientists and to do the experiments in the lab now. So the industry, actually is will be we think will be the next generation of the industry, and we have already 4 waves of the evolution of the industry, and the 5th will be, we think, will be the synthetic biology. What's synthetic biology? I don't go to the details, and this example, and the synthetic biology has become really important, or also they become it's unbelievable fast, especially to define some rare disease. Is, for example, this COVID-19, and after we got the sequence of the COVID-19, and by using synthetic biology, we just spent 1 month to get the COVID-19 already rebuilt or reconstructed in the lab, or in the to have this synthetic COVID-19 virus, and for the study and for the drug evaluation. So this is one example of the the synthetic biology can do really a good job, and much faster than the before. So synthetic biology nowadays not only in the medical department, medical part and field, and we also have a lot of since 2008, 2003, and the first E. coli engineered E. coli cells can produce artemisinin, and this is our first synthetic biology example. And since then, so nowadays, every year we have many synthetic biology example coming out. And the last one is in 2020, and the collagen produced the lactoferrin by microbial fermentations. So this is a development of synthetic biology, and as the former speakers already mentioned, the example of synthetic biology enterprises. The first one is the medical, in the medical field, and Moderna, and this is a company has market value of 62 billion, and they are developing mRNA drugs and vaccines. It's really successful, and it got a lot of investors to invest their technology and their research and development. And as everyone knows, the vaccine innovation is one kind of a synthetic biology innovation. And in the past, most vaccine takes years to develop. But nowadays, science can create multiple vaccines especially from the SARS COVID-19 within a year, actually shorter than 1 year. The larger example of synthetic biology enterprises is food, and so the former speaker said already the impossible biotech company can build, actually used to develop the artificial meat, and this is one kind of development to save our population, big population, from the short of the food. As those impossible investors include many famous directors of the companies, and several now here, including Bill Gates. So the unfolding of synthetic biology industries become wider and wider, including this not only the medicine and also now the food, agriculture, and the chemicals, software, and the DNA synthesis, RNA synthesis, etc. So the synthetic biology industry will lead the boom of the bioeconomy. As Mankizen said, the market of the 500 billion with a compound growth rate of 28%. The whole biotechnology field will bring a direct economic output of the 3 to 5 trillion annually. So this is impossible growing of the biotechnology and synthetic biology. Of course, many countries around the world are planning the development of synthetic biology, including USA, UK, EU, and also especially China. Our country, China, attached great importance and places great emphasis on synthetic biology. Our Chairman Xi has many speeches focused on the biotechnology. For example, he said the synthetic biology, genome editing, neuroscience, and regenerative medicine, and so forth, which will represent life science, are incubating revolutionary progress. So based on this leaders emphasize, and the Chinese, most of the Chinese Ministry of Science and Technology already announced the program on the synthetic biology. And based on that one, the Shenzhen city also combined with the most has put, I think it's around 1 billion of Chinese yuan on the synthetic biology to promote the synthetic biology basic research and also the application. So therefore, the Shenzhen city, in the Shenzhen city, we have in China, we have first institute of synthetic biology. This is already since built up the on 2017, end of 2017. Now it's 4 years now, and Shenzhen city also matched the funding to the Institute of Synthetic Biology. And we have the world first synthetic biofoundry, and it's located in the Guangming district in Shenzhen. And the buildings will be going to ready this year, and we can move in next year. So the world first faculty of synthetic biology is also located in our Shenzhen Institute of Advanced Technology. The distribution of domestic talents in synthetic biology in China has focused on Beijing, Tianjin, Shanghai, and Shenzhen. Actually, as you can see, the Shenzhen's become very important center of the synthetic biology, and we have the world largest synthetic biology research team, and we over 800 people. We have 43 full-time PI, which they are all very young, and this is our the university, Shenzhen Institute of Advanced Technology, and we have actually the college for synthetic biology, or goal for synthetic biology is fostering talents, promoting scientific innovation, and serving the industry. So the synthetic biology college and has already the director in position, and we're going to achieving the building a global innovation network system in Shenzhen. So the Chinese first Shenzhen Symbio Association is already is affiliated with SAT. So we also have a CCIC. This is similar to IGM, and to promote the young scientists or young students in China to understanding or to perform the synthetic biology research. And we also have a synthetic biology specialized committee, and we have a very large team in China to perform the synthetic biology research. Another one is the Asia Synthetic Biology Association. This is located, the headquarters is located in Shenzhen, and we have we have 12 nations and co-organize this global biofoundry alliance. So biofoundry plus research institute push for 0 to 1 project. But the proposition is how to realize the touching products along the way. So therefore we have the Shenzhen city actually build a really innovation and enterprise complex building for us. So we have the upstairs and downstairs activity, and upstairs we do the innovation research, and downstairs we have the newly set up the companies to perform to transform those innovation result. So we have already 25 resident companies, and there are 13 high-tech enterprises ready to move into the biofoundry, and the 12 enterprises are being selected. So those enterprises in Shenzhen Institute of Advanced Technology, we include in those like synphage, sensors, biosciences, Beijing, and genome, palm, and sea atom. So they are all doing the synthetic biology technology, Geoff technology, also the products. The first example is sensor biosciences, and they are doing the construction of artificial yeast, realization of the cannabinoid biosynthesis for the first time. And this is we got already these sensors already got 20 million of angel fund, and their value is already to 100 millions. The second example is realizing the storage of the random data this information in DNA. As everyone know, nowadays the data storage are using the chips, but actually we can use DNA to do the data storage. One sample size of the DNA can store one national library data. So this is already performed by sea atom, and they already got the 20 million angel fund, and the value is already also is 100 million now. The third example is the development of the glue, the living glue, and this is by Palm Biotechnology, and actually we are using the biological glue to do the many surgery repairment. So this is a the Palm has a 5-year capital, and there's already 5 million of angel fund investment, and the one is already 27 million now. So the upstairs and the downstairs stories is already listed as experience of national development and the reform commission, and as an example to promote this stairs in China. So the sorry. So the with the biofoundry innovation system, we are trying to build a wide range of application scenario by project practice by using 3 methods. The first one is the proof of concept center. This is the source to prototype. The second is upstairs and downstairs. This is from prototype to products. The third one is specialized industry part. This is from products to mass production. Actually, this is from 0 to 1, and 1 to 10, and 10 to 100, and 100 to the mass production. This from basic research application to the applied research. So this is a university plus research institute and plus biofoundry. So synthetic biology, synthetic biology, we have also the funds, and Shenzhen has amalgamation of opportunities for the synthetic biology industry, and taking the advantage of its innovation from the source to empower the synthetic biology industry. So the fund is built by 3 parts, as is university and the private investment groups, and also the city from Shenzhen. So we have a lot of data analysis platform. This is also the to engineer the biological industry to actually get them insights and to promote this biotechnology, synthetic biology. And we have engineering. We have engineering the in Shenzhen, in Guangming Science City, we have very large the engineering committee. Zip it around here. Sorry. Now we are going to gather in all this synthetic biology in the Science City, and we have 3 departments, 3 parts. One part is our university, the Shenzhen Institute of Advanced Technology, and the second one is the biofoundry, and this is also including the College of Synthetic Biology, and the third one is the Engineering Biological Industry Innovation Center. So as I said, these 3 parts together will form the basic research to the applications, and actually there's a top loop of the synthetic biology. The first part is the research, focus on the research and the technology innovation, and we have 5 in one. For example, research institute, education, colleges, industry, and special subjects. And then the larger part is the industry incubation, There's also 5-in-1, like for example, industry insights and the roadshow industry foreman, and also with capital angel funds, and to form together and to develop the hatching products along the way and transform the on spot. So in the conclusion, so the in China, the global is in the synthetic biology and the industry pattern. So everyone can see the numbers of the research paper are increased massively. Also, the number of synthetic biology enterprises increased massively in China, and you can see not only in China, also in globally, and there are more and more synthetic biology industry companies are created. So Shenzhen now in China has a biological industry is only 2%, and it's very low at this moment. And but we think, or we predict the biotechnology, especially the synthetic biotechnology, will become the major or main industry in Shenzhen in the near future. So the waves of the industry in China, especially in Shenzhen, will become the third one, and Shenzhen will become the synthetic biology industry center in the future. So this is all, and thank you for listening.